Hi, so today I'm speaking about geometric constraints on human brain function, which is primarily based on our publication that we had about a month ago, which you can scan via the QR code if you want to know more about it. So the paper and the talk, we will be focusing on trying to answer the question on how does the underlying anatomical structure of the brain constrains its emerging dynamics and function. So we can start from the beginning. The conventional view is that because the brain is such a multi-scale organ, we can construct different scales of networks or connectomes from the nanometer to the macro millimeter to centimeter scales, in using quite different techniques such as diffusion MRI and so on and so forth. The limitation of this network view is that it cannot fully explain a lot of the different emerging phenomena that we've seen in different data sets in both human and non-human models. Say, for example, in mouse, using optical imaging, you can see this propagation of very, very continuous wave-like pattern in its brain. So a class of mathematical models that can capture this continuous manner of propagation is what we call neurofield theory. Basically, neurofield theory approximates the activity of populations of neurons, much like how a group of starlings could move, even though each bird would have its own activity. And neurofield theory heavily relies on the connectivity between these populations that are connected by a kernel W, which is focusing mostly on very, very local and exponentially decaying activity. That is, the weight of activity decays exponentially as a function of distance. And most importantly, neurofield theory approximates that the firing rate of a population can affect another population to give rise to its activity following this damped wave equation, much like how when you throw a pebble into a pond, the activity will propagate, but will exponentially decay in space and time. And the important bit here is this spatial kernel, the Laplacian, which states that the physical geometry of the substrate, basically here, for example, the cortex, is actually pretty important, and it matters a lot. So we have these two viewpoints, and we have to try and ask which structural feature of the brain do we use to better study the macroscale dynamics we observe, say, in fMRI. On one hand, we have this connectome view, where you discretize the cortex and you prioritize node-to-node dynamics and the highly complex connectivity between these nodes. On another node, you can focus on the geometry or the shape of the brain, where you treat, say, the cortex as a continuous entity, and you prioritize wave dynamics, space, geometry, and very, very local and EDR connectivity. Now, how do we capture these structural constraints? We adopt methodologies from physics and engineering, where for a particular system, you can capture what we call as the eigenmodes. And these eigenmodes are the natural spatial frequency patterns of vibration or excitations of a particular system. So, for example, for a 1D string, you would obtain this family of eigenmodes that have very, very unique patterns. And in a 2D cladding plate, for example, each plate would have its own intrinsic eigenmode. And the properties of these systems, the string and the plate, heavily dictates the patterns or the eigenmodes that you will get. When I say properties, it could be the length of the string, the density of the material, and the length, the size, and so on and so forth. Now, the important aspect of these eigenmodes is that they are what we call as a basis set. So they are a complete basis set. That means the eigenmodes are orthogonal. There's no overlap between them. And that means you can perform a mathematical exercise called the basis set expansion, where any signal can be approximated as a linear combination of eigenmodes psi weighted by some coefficient A. And you can do this exercise for any kind of signal emerging in that system. So now, how do we calculate the eigenmodes of the brain? So we have this cortical surface mesh approximating the cortical geometry and a connectivity matrix approximated from diffusion MRI. We do that by solving this Laplacian eigenvalue problem. It's like an eigen decomposition, where this is what we call the Laplace spectrometer operator, which encodes the spatial relationships between points and nodes. And you can get a family of eigenvalues lambda and a family of eigenmodes psi. And depending on the problem that you're trying to solve, you can approximate the Laplace spectrometer operator in quite a variety of ways. We can discuss that later on if you're more interested to know more about that. So we calculated 3 different sets of brain eigenmodes. The first 2 will be the geometric and the connectome, but we also added 1 bit, the EDR, the exponentially decaying rule connectome. Basically, here we create a synthetic network following a very, very stochastic wiring process with a probability that follows an exponentially decaying rule. Again, in agreement with a lot of observations in human and non-human species, where weight decays as a function, as an exponential function with respect to distance. By doing so, we can get these 3 sets of eigenmodes, which are arranged in a very, very peculiar manner, where the first mode would correspond to the to modes with low spatial frequency or long spatial wavelength, and the higher modes have higher spatial frequency or short spatial wavelength. So there you see here the activities are very, very localized. And then we use these eigenmodes to study empirical fMRI data from the Human Connectome project, both resting state and task-evoked. And what we found is that, say, the data here on the left can be reconstructed by the geometric modes just by using 100 of those. And you can see that the accuracy sits on about 80%. This is the accuracy as a function of the number of modes, and we're sitting here at 100. And if we compare the geometric eigenmodes with the other anatomical basis sets, and again, I have to emphasize, we're only focusing here on basis sets derived from the anatomy of the brain, nothing else. We see that the geometric modes seem to perform very, very well as compared in both resting state and task activation maps, but the EDR modes seem to perform well as well, suggesting the importance of this EDR-like connectivity kernel. And then when we ask, okay, how far can we go? Because remember, the geometric eigenmodes were based on neurofield theory, which uses a simple wave model. So we asked, how far can we go with this isotropic wave model? And we did that for both resting state and stimulus-evoked activity, and compared it with a highly sophisticated neuromass model that relies on a connectivity matrix. And what we found is that, irrespective of the metric you focus on, we found that the geometric modes plus wave model either perform as well or better than the highly sophisticated neuromass model. And when we did the stimulus-evoked activity task, it seems like we can get these hierarchical temporal responses. And then if we follow through it across the visual hierarchy, we find that it strongly correlates with non-invasive measures of hierarchy like the T1, T2 data. And then we use these geometric eigenmodes to study quite a different number of maps, because in traditional brain mapping approach, you have a map and you threshold it and to get isolated clusters signifying where the activation mostly focusing on. But what we're showing is that if you actually analyze the unthresholded maps of HCP and about 10,000 maps from NeuroVolt, we see that the power sits in those modes that have very long wavelengths. That means it kind of covers the entire brain. And if we look at how we can reconstruct the data in both ends of the spectrum, if we keep removing the long wavelength modes, that's the solid line, as opposed to the short wavelength modes, the higher modes that we have, that's the dashed lines, we see that you can impair the reconstruction accuracy much more faster if you remove long wavelength modes, suggesting that indeed empirical data has this long spatial component to it. And finally, we employed the same approach in the subcortex, and we calculated the eigenmodes of the three-dimensional volume, three-dimensional volumetric shape of the thalamus, striatum, and the hippocampus. And what we found is that the geometric modes of these subcortical structures almost have perfect correspondence with functional gradients derived from fMRI data, which is quite intriguing, because again, the modes were just derived from the three-dimensional anatomy of these structures. So what are the advantages of this eigenmode-based approach? One is that it provides a more physical understanding of the relationship between structure and function. The eigenmodes can easily be calculated from T1 weighted images, which most neuroimaging studies have. That means it can be robustly and flexibly applied across different contexts, both in humans and actually non-human species. So in summary, what we found was that cortical geometric eigenmodes can be used to study the anatomical constraints on brain function. And we have to emphasize that our results does not show that connectivity is not important. The geometric modes has its own implicit connectivity, but what it's saying is that the contribution of complex connectivity beyond the local media may be more subtle, and their influence could be more observable at resolutions not captured by fMRI. We've also shown that traveling waves can provide a really simple but highly efficient mechanistic account of data and physiological phenomena, and that task-evoked activity in fMRI is dominated by these long wavelength modes, and that the geometric constraints that we found are pretty universal, both because we can find in both cortex and subcortex. And most importantly, the work opens up quite a variety of new opportunities for mapping healthy and diseased brains, and both in aspects of neurodevelopment or maybe even in evolution. And I just want to acknowledge all my fellow co-authors. And again, you can scan the QR code if you want to know more about the paper. Thank you.